because our restream is getting set here. Okay, and we're now live. Starting to let everyone in. Good afternoon, everyone. Thanks for joining us. We're going to give it a couple of minutes for everyone to come in the room here. Good afternoon again, everyone. We'll give it about another minute and a half here to fill the room up. Good afternoon, everyone. Thanks again for joining us. We're going to give it about another 30 seconds to fill the room. Great. Well, good afternoon, everyone. Thank you once again for joining us for our weekly Wednesday webinar series. Today's webinar is on monitoring the coagulation status of people with hemophilia receiving Emicizumab. My name is Brett Spitali, and I'm the Vice President of Advancement here at the National Hemophilia Foundation. At any point during the webinar, if you'd like to ask a question, please use the Q&A feature at the bottom of your screen. We have NHF staff members monitoring these questions, which we will pose to our presenters after the presentation. This webinar is being recorded and will be available to the community beginning on Friday, October 22nd. Today, we'll also be live streaming on our LinkedIn and YouTube channels. Today, I'm joined by Dr. Hande, and Hande, I apologize, I don't want to butcher your last name here, but Kizilocak, if I'm close.
Yeah, close. And yeah, you're super close.
Great. She joins us out in Los Angeles, and Dr. Guy Young, who is currently flying over Indiana somewhere, joins us as well too. And Dr. Valentino is also online with us as well. Thanks for taking the time to join us today, and Hande, I'll turn it over to you now to get us started.
Thank you, Brett. Good afternoon, everyone. It's my pleasure to present our study about determining the factor VIII equivalency of emicizumab by thrombin generation assay in patients with severe hemophilia A. And I would like to thank the National Hemophilia Foundation, Dr. Valentino, for his kind invitation for this talk. Okay. And as we all know, hemophilia A is an inherited bleeding disorder characterized by the deficiency of coagulation protein factor VIII. And over the past 50 years, hemophilia patients and caregivers have witnessed great advances in the treatment of this disease. And this success has been achieved by the modifications of therapy and especially with the development of novel agents and more tailored therapy options. And one of these novel agents is emicizumab, which is a humanized bispecific monoclonal antibody, and it bridges activated factor IX to factor 10, and this way it mimics factor VIII, but the molecule itself has no structural homology to factor VIII. And safety and the efficacy of emicizumab has been evaluated during the Haven clinical trials, and it has been shown very effective for bleed prevention in hemophilia A patients, and the annualized bleeding rate has been reported to be below 1 for all of the patient groups, that is patients, that is adults and pediatric patients with and without inhibitors. So what remains unknown in the area of this new drug is the degree to which emicizumab corrects the coagulation defect. So conventional assays, which are APTT and one-stage clotting factor assay, cannot predict the factor VIII equivalency when there is emicizumab in the body. because APTT is normalized already at subtherapeutical emicizumab concentrations, which is around 3 to 5 microgram per ml. However, the therapeutic emicizumab concentration is approximately 10-fold higher than that, which is 50 microgram per ml. And on the other hand, human chromogenic factor VIII assay can monitor the emicizumab level, and as you can understand from the name of the test, it contains human factor IXA and factor X. And since emicizumab interacts with human factor IXA and X, this test can monitor the emicizumab level, only the drug level, and still these values do not relate to a true factor VIII equivalence. So the important point is, why is it crucial for us to know the factor VIII equivalency of emicizumab in patients with severe hemophilia A? Because as you all know, patients on emicizumab, albeit infrequent, can still experience breakthrough bleeding. And also, it's important to manage the patients when they go through a major surgery. So for this reason, it's important to know the factor VIII equivalency of emicizumab to further manage the patient, especially during a breakthrough bleed or when they go under major surgery. So for this reason, scientists conducted an in vitro study, and in this study, they used a sequence-identical analog to emicizumab, and they drew blood from patients with severe hemophilia A, and then they spiked this sequence-identical analog into plasma samples from these patients in vitro. And then they tested APTT. They tested 5 different commercial assays for factor VIII chromogenic assay. And also they ran clot waveform analysis and thrombin generation assay. And as I mentioned in the previous slide, the authors also concluded that APTT and factor VIII chromogenic assay can lead to false, low, or high results and cannot be used as a tool to predict the factor VIII equivalency of emicizumab. And not only the conventional assay, but the thrombin generation assay has led to discrepancies in the results. And it depends on which technique they use or which method they use for thrombin generation assay. Because you can see here, when the assay used tissue factor trigger instead of factor XIa trigger, factor VIII equivalency of SAA was found to be approximately 3 times higher. And the peak thrombin with SAA, when triggered by factor XIa, even at the concentration reported as therapeutic range for emicizumab, only reached to 4 to 8% as a factor VIII equivalency. So this in vitro study emphasizes the need for standardization of thrombin generation assay when used with emicizumab. And I'm going to focus on the standardization later in the talk as well. So after this in vitro study, there have been 3 in vivo non-human primate studies. And in the first study, primates were given an anti-factor VIII antibody to mimic the hemophilia. And then they were given either emicizumab or porcine factor VIII. And the reason that they chose porcine factor VIII is it's not going to be blocked by the anti-factor VIII antibody. And then both treatments were compared by prevention of blood loss. And they did this by consequent measurements, by having consecutive measurements of hemoglobin levels. And emicizumab activity at these concentrations corresponded to 1% of factor VIII activity. Since they used 6 microgram per ml for emicizumab, and since the therapeutic concentration is 10-fold higher, they concluded that the factor VIII equivalence of emicizumab will be around 10%. And another in vivo non-human study used the same primate model, and they concluded the same result as the first study. And the third study used a factor VIII deficient mouse model with an addition of human factor IX and X, and they used the tail clip bleeding model. And after the initiation of the bleed in the tail, the mice were given different concentrations of factor VIII, and a calibration curve was created with specific factor VIII concentrations correlated with specific amount of blood loss. And then the reduction in blood loss in therapeutic levels of emicizumab corresponded to a factor VIII concentration around 9%. So all of these in vivo studies have confirmed the notion that patients with severe hemophilia A on emicizumab have a bleeding phenotype similar to mild hemophilia. And to further elucidate this factor VIII equivalency of emicizumab, we conducted the first in vivo human trial. And in our study, our objective was to generate a calibration curve from the factor VIII levels from patients with mild and moderate hemophilia A and also normal healthy controls and their thrombin generation parameters. And we used two parameters. One is underground thrombin potential, ETP, and the other one was peak thrombin, with the objective of determining the factor VIII equivalency of emicizumab by thrombin generation in patients with hemophilia A and inhibitors on emicizumab. And now on in the talk, I'm going to use the acronym factor VIII EMT for factor VIII equivalency of emicizumab by thrombin generation. And we've enrolled 25 patients with moderate and mild hemophilia, and none of the patients were on prophylaxis, and none of them have infused any factor or drug, at least in the last week prior to the blood draw. And they were all on non-bleeding states. And we enrolled 11 patients to the severe hemophilia A group, and all of the patients have been on emicizumab at least for 6 weeks for the study level. And none of the patients have infused any factor or bypassing agents at least in the last week prior to the blood draw. And we enrolled 4 patients to the healthy control group. And for thrombin generation, we collected the blood samples into S-monovet tubes, and these tubes were preloaded with contrypsin inhibitor. And so the reason that we chose to use contrypsin inhibitor is once the blood touches the tube, the contact activation starts. So contrypsin inhibitor is blocking this by blocking factor 12, which is the contact factor. And this way we avoided the extra thrombin generation in the tube. And then we prepared plated pool plasma, and we stored the samples at -80, and then we ran the samples in batches. And for the trigger, we used 1 picomolar of tissue factor, and also we chose to do the warming for 10 minutes as the incubation period. So all of these steps are important when we ran the thrombin generation assay, because for now it varies between laboratories, and so different laboratories are using different techniques, which leads to discrepancies in the results for thrombin generation assay. And that makes it crucial to have a standardization for TGA, so that in the future we can, when we use it more widely, and we will have more accurate results, and we will avoid the discrepancies, the interlab discrepancies. And for factor VIII activity, we use the one-stage APTT-based clotting assay, and we ran the factor VIII activity for mild and moderate hemophilia patients and healthy control group. And if you go back to the patient's demographic table, so we enrolled 25 patients to moderate and mild hemophilia group. However, one of the samples wasn't sufficient, so we analyzed 24 samples from this group. And we enrolled 11 patients to severe hemophilia A group, but then one of the patients moved out of state, so we analyzed 10 patients here. And the median age was 15 for the severe hemophilia group, with a range from 4 to 33. So first, we created a regression line And we used the factor VIII activity results from mild and moderate hemophilia A patients and the healthy control. And then we added the ETP levels of these patients for creating the regression line. ETP is the endogenous thrombin potential, and it is one of the parameters that we use from TGA. And then once we created the regression line, we plotted the ETP levels of the patients with severe hemophilia A to this line. And as you can see here, 3 of the 10 patients had a factor VIII EMT between 10 to 20%, and 4 patients had a factor VIII EMT between 30 to 40%, and 3 patients had a factor VIII EMT about 60%, which is already in the normal range. Then we generated the regression line, this time with another TGA parameter, which is peak thrombin. And again, we use the factor VIII activity results from mild and moderate hemophilia patients and healthy controls. And after generating the regression line, we plotted the peak thrombin levels of patients with severe hemophilia A on emicizumab. And as you can see here, 3 of the 10 patients had a factor VIII EMT between 10 to 20%, and 4 patients had a factor VIII EMT between 25 to 30%, and 3 patients had a factor VIII EMT between 35 to 40%. So these results are also confirming the previous in vitro studies and also in vivo non-human primate studies and also the observation that we've seen in clinic as a patient with severe hemophilia A on emicizumab have a bleeding phenotype similar to mild hemophilia, because all of our results were about 10%. The patients are either in the mild hemophilia range, or some of them are in the normal healthy control range. And another interesting thing we observed during the study is factor VIII EMT was tightly correlated to a weight, with the heaviest patients having the lowest factor VIII EMT. And we also analyzed the factor VIII EMT by age-adjusted BMI category bar per CDC guideline. And as you can see here, we ended up with the same result. Overweight or obese patients had lower factor VIII EMT compared to underweight or normal patients. And this may be related to a volume of distribution of emicizumab, and suggests that overweight or obese patients may have some drug distributed outside of their plasma compartment. And this issue has also been evaluated from the database of the Haven clinical trials. And a population pharmacokinetic model was developed with 389 pediatric and adult patients from the Haven database. And the potential baseline covariate effects were assessed, which are body size, age, race, presence of factor VIII inhibitors, and albumin levels. And then the study demonstrated that body weight was strongly positively correlated with clearance and the volume of distribution. And then they concluded that this positive correlation between body weight and the clearance and the volume of distribution represented the catabolism of the antibody, which is emicizumab, by the reticular endothelial system in reticular connective tissue, which is widely distributed throughout the body. And if I go over our limitations of our study, first of all, we enrolled small number of patients from a single institution, and we only used one TGA measurement method. And as I have mentioned previously, different methods using different triggers or different methods, as they can skip the incubation period, or they can end up using another tube than as Monovet tube, or they can choose to skip the CTI, which is a contraception inhibitor. So all of these variables could yield to different results, although we use the same method to generate the calibration curve, and this should mitigate against this issue. And we also measured the factor VIII activity by the one-stage APTT-based clotting assay, and we did not perform the factor VIII chromogenic assay. Also, we did not measure emicizumab plasma levels, and we cannot correlate the results with the actual emicizumab levels, which probably could have been useful given the findings of different factor VIII EMT, the lower factor VIII EMT in the overweight or obese patient group. And last of all, as there were very few bleeding events in the inhibitor patients in this study, we were unable to correlate these results to any bleeding outcome. As a conclusion, understanding the degree to which emicizumab corrects the coagulation defect is an important goal as it has clinical implications. As I said, it's important to manage the patients, especially during breakthrough bleeding episodes or if they go under major surgery. And the data from the study suggests that hemophilia patients on emicizumab function like patients with mild hemophilia. As you have seen in the results, the factor VIII EMT fell within the range of 10 to 40%. And this has been also presented on the previous animal in vitro studies. And also it is supporting the notion that we see in the clinic as patients act like they have mild hemophilia with their bleeding phenotype. And for sure, additional studies are needed to confirm these findings and especially needed to determine if this unexpected outcome of lower factor VIII EMT in overweight or obese patients is a true relationship. And our study was funded as an investigator-initiated award to Dr. Young from Genentech. And thank you for listening, everyone. And we would be happy to answer if there's any questions from the audience.
Thanks so much, Hande. We appreciate it. We do have a couple of questions that have come in, and let me go up here and scroll through and start. First question is, I noticed 80% of the participants were Latinx. Does this pose any issues in applicability of the data to other ethnic groups?
Or 80% of the patients is?
Latin.
Can you repeat the question? Oh, okay. Yeah. So as I said, we have enrolled like a small group of patients from our hospital. So it might, but to further evaluate it, we need to have like more studies involving more patients from and from probably like from different institutes, so we can see if this is an effect for the results.
Great. Another question that comes in. You mentioned the limitations of your study, but what were the greatest challenges during your data collection and analysis?
Yeah, so for the so I can say since patients on emicizumab, so they don't have like most of our, actually not most of, but all of our patients were on a non-bleeding state, and so sometimes it's hard to like do the blood draw, or some of the patients have their ports removed, so it was hard to get the blood draw once their port is removed. But luckily they were happy to participate in this study, and they helped us to further evaluate this and help the hemophilia community. So I can say, yeah, maybe the removal of the port was a challenge, and also the patients were in non-bleeding state, so they were coming only for their regular visits at the clinic. But again, I appreciate all of them participating in this study.
Great. And Dr. Valentino, I don't want to butcher this too bad, so correct me if I'm wrong here when I ask this, but this came from the audience. Why is there a difference in thrombin generation between TF and is that factor 6A activation?
11.
11A, I'm sorry, 11A activation, yeah. So I'm sorry, Hande, that question was for you. It was.
Oh, for me? Oh, sorry.
Yeah,
Yeah,
I
I thought it was
apologize.
for Dr. Valentina.
No,
Okay.
I was just
Yeah.
asking him for an assist there if I needed it, but that one's for you.
Okay. So when we run the TGA with tissue factor, we can actually use like two different concentrations. So it can vary in like under the TF2. So we can run it either with 0.5 picomolar, or we can run it with 1 picomolar, and we can run it with factor 11A. Since factor 11A starts the coagulation, like starts the thrombin generation from the intrinsic pathway, it creates more thrombin bursts during the thrombin generation, and it ends up with a higher result, kind of. And also it also changes when we run the samples with 0.5 or 1 picomolar of tissue factor 2. But most of the studies are using, and most of the labs are using tissue factor as a trigger. So that's the reason we chose that. And also the latest guidelines from IACH has supported the use of tissue factor 2. But still, different labs are using either factor 11A or tissue factor. So that's the reason we have to have a standardized thrombin generation assay to avoid this discrepancy. So it depends. It basically depends on that tissue factor is coming from the extrinsic pathway, and the factor 11A is activating from the intrinsic pathway, and it's creating like different thrombin generation levels.
Great. And Dr. Young is on, and he is answering some questions in the Q&A here, and I'm hoping everyone can take a look and see that. But I'm going to ask this one, even though Dr. Young had answered it. He had said your efforts are Prasad asked your results differ from Peter Lentine's article, which showed factor 8 equivalency of about 9%. Any thoughts why this may be? And he also commented and said, "This is great work."
So that question is for me too, right, Brett?
Yes. Yeah. I'm sorry.
Okay.
Okay. Yep.
Yeah. Okay. So Dr. Lentine's study has concluded. So first of all, it was a non-human study, and it was an in vivo non-human study, and they concluded a factor 8 EMT around 9%. So non-human studies can vary from human studies. So that was the the biggest thing that drive us to conduct a human study. And the first and the biggest difference is they used a mice model, which we ran the samples. We got the blood draw from patients currently on emicizumab, and like spiking emicizumab into a blood sample versus having the blood from an actual human on emicizumab, it's totally different, and it's probably the biggest reason of the difference, and that makes human in vivo studies more maybe more close to real life results. But still, we concluded the same result with them. So they found the factor 8 EMT around 9%, and our lowest result was around 10%. So basically, we all concluded that at least patients are acting as if they have mild hemophilia. But as I said, the biggest difference is they use the mice model, and we are doing the in vivo human study. So it takes into account like different coagulation happening in the body with emicizumab versus the tail bleed model in the mice.
Great. And we have a comment here, and I wanted to give you the opportunity to comment on it. But it says, interesting. When I first started Hemlibra, the idea was I would not bleed, but now we are getting closer to the idea of I bleed more like a mild, which seems to be borne out in my experience. Any comment on that at all, Hande?
Yeah. So also I mentioned that we haven't seen like bleeding events in our patient group, and we couldn't correlate our results to any bleeding outcome. So the comments that we are making on is not actually about the patient's bleeding phenotype. So we are concluding results from laboratory results, and so this doesn't necessarily mean that they're going to have, like, the patients in emicizumab are going to have like bleeding episodes as mild hemophilia patients, because we haven't seen any bleeding events like in clinics, and I think most of our patients are in the normal range too. So the lowest factor VIII EMT was in the mild range. So it doesn't necessarily mean that all of the patients are going to have the mild bleeding phenotype. And yeah, so at least close to half of our patients were in the normal range. So hopefully we'll conduct studies with more patients, and then we are going to have more accurate results, and maybe we can find out that it's more close to the normal range. But as I said, in clinic patients, we don't see any bleeding events in the patients. And the important part is we still have to manage the patients when they have a breakthrough bleeding, or especially when go under major surgery. So we all know that we have to have, like, most of the time we have to add bypassing agents to the bleeding prevention therapy. Yeah, so it doesn't necessarily mean that patients are going to bleed like mild hemophilia patients in real life.
Great. Thank you, Hande. Next question that came in, what will be the next steps in your research to build on the findings of this study?
Sorry, I lost your voice. Can you?
Oh, yeah, sure. I'll slow down a little. I have a tendency to talk a little bit quickly. It said, what will be the next steps in your research to build on the findings of this study?
Yeah, so we actually had another study since we showed that patients are having a mild hemophilia phenotype, and we had another study which we ran at the same time with this study, and we had plasma samples from patients on emicizumab, and then we spiked different concentrations of bypassing agents into these plasma samples so that we can find the safest bypassing agent level to use on patients on emicizumab if they go under major surgery or if they have a breakthrough bleeding event. So we did that study with thrombin generation assay too, and also in the future we are going to have another study to validate the accuracy of thrombin generation assay, and we can compare the in vitro versus in vivo levels. Since the other question from the audience, they asked that, like, why our results are different from Peter Lanting's study. Our results are close to, but not exactly the same. So they're going to compare the in vitro and in vivo thrombin generation assay in not only in patients on emicizumab, but in patients with different hemophilia levels and on factors, or on extended half-life factors or standard half-life factors on emicizumab and on other novel agents. So we are going to compare the in vivo to in vitro results, and we are going to further evaluate the thrombin generation assays accuracy too. And also, it would be better to conduct the study with more patients, and it would be better if we can run the factor VIII chromogenic assay, and if we can run the emicizumab levels so that we can compare the results. And hopefully we're going to conduct a study including these parameters too.
Great. Thanks, Hande. We've got one comment and one question that have come in, and I'll read the comment first and allow you to respond to this. As a severe A, I had lots of breakthrough on 80% prophy. On Hemlibra, I'm doing a lot less and very few major bleeds. I didn't know if there was anything that you wanted to comment on that.
Yeah, that's great to hear that emicizumab is, making the helping with the bleeding episodes, and yeah, I'm happy to hear the outcome.
And then another question that came in, when the subjects were recruited, what efforts were taking place to ensure diversity?
Yeah, so at our clinic, we have pretty much like a diverse population. So we reached out to all of the patients that we had at the current time that we were conducting the study, all of the patients with inhibitors on emicizumab, and we enrolled all of the patients that were on emicizumab at that time. And as I said, we would be happy to conduct another study so that we can enroll more patients from more centers so that we can have a more diverse population. But as for our hospital, we enrolled all of the patients on emicizumab at the time of the study. And again, I'm thankful that all of our patients have concluded to this, help us to conclude these findings so that we can help the hemophilia patients, and we can help scientists to conduct future studies.
Great. And we have a question that came up, and I know it looks like Dr. Young is typing out an answer, but I'll ask here as well too, Hande, if you don't mind weighing in. I'm still trying to understand the dosing of Hemlibra when a patient experiences a trauma or has an upcoming surgery. How do you know how much factor to give a person in these circumstances? Does the amount change depending on how long it has been administered? They've been administered Hemlibra. And I can reread that if you'd like me to.
Yeah, so if the patient is on emicizumab, if it's at least for 6 weeks, then that means the patient has a steady emicizumab level. For that question, so how do we know how much factor that they're going to give or how much bypassing agent that they're going to infuse to the patient? That was the reason that we ran the study so that we can tailor the therapy by running thrombin generation assay on a patient on emicizumab, so that they're going to have their baseline thrombin generation level only with emicizumab. And then when they have the bleed, we will know how much factor or bypassing agent we are going to give the patient.
Great. Thank you so much. And we have one more question. It looks like that has come in, and might be a good one to end on here, unless something else comes in. But what would be some of the clinical recommendations as a result from your study?
Sorry, Brett, can you repeat?
Yep. Sure.
Did you read a comment or a question? Could you repeat it? Sorry,
it was, yep, it was a question, and it stated, what would be some of the clinical recommendations as a result from your study?
Yeah, so as already shown in having clinical trials, emicizumab is very effective for bleed prevention, but still, since we've figured out that patients are acting as if they have mild hemophilia, that means they might or might not, but they might need extra factor therapy, like a bypassing agent. So if that's the case, and if the center that's following that patient is able to run thrombin generation assay, that would be very important to manage the patient's bleeding. And also they can use the regression line that they have created so that they can have an idea of the patient's factor VIII EMT on emicizumab, which will help for the future management of the patient. But still, there's like we have to run like more studies. We have to conduct more studies with more patients to further evaluate this.
I'm not seeing any more questions or comments that have come in, so I just want to take a minute. Hande, Guy, and Len, thank you all for joining us today. Hande, thank you for presenting the information. We truly appreciate it, and I know this is
my pleasure.
Yeah, this is a very, I think a lot of people are anticipating this and excited about it. And you can tell by the attendance. Our attendance is wonderful today. So we really appreciate you taking the time out of your busy schedules to join us, and you in particular as well, Guy, joining us in the air. We really appreciate that. Um, Everyone, please note that this recorded webinar will be available on Friday, October 22nd at hemophilia.org under the events tab with all of our other archived webinars. You can also find it on our LinkedIn and our YouTube pages as well, as it'll be archived on there. Again, I'd like to take the time to join or to thank our panelists, and we truly appreciate your joining us and taking the time to do that. Everyone have a great day. Thank you so much.
Thank you.